So I will move to medicinal mushroom and effect of an environmental factor on the quality of the benefit activity of the beneficial of the medicinal mushrooms. As said, I work in MIGAL, Galilee Research Center and Tel Chai College in Israel. We focus on biotechnology, agriculture, environment, food, and nutrition. And my specialty is edible mushroom. As you see here, I was working on pleurotus, on shiitake, on agaricus, and now my research is focused on morel, morchella. I assume that many of you know it. So what I'm going to talk about is the effect, is the antioxidant activity of medicinal mushroom and what affected a little bit. Why it's important, we all know oxidative stress may cause a lot of diseases in the human being. Oxidative stress is actually the production of reactive oxygen species which attack DNA, attack organic compound. This, of course, this damage cause also all the diseases. As we see it here, it's from the brain, Alzheimer, Parkinson, memory loss, depression, stroke, trauma, all this is the result of oxidative stress in the eye, in the heart. Many of the problem of heart diseases are caused by oxidative stress, of course, lung, asthma, in the skin, psoriasis. Oxidative stress is very important to the health of the people. Oxidative stress is, again, I will say it's the production of reactive oxygen species. These compounds are attacking the DNA, attacking organic molecules, and because of that, there is damage and diseases in the human being. As I said, it's starting from the brain, Alzheimer, Parkinson, memory loss, and so on. In the heart diseases, lung, asthma, for example, is caused by this oxidative stress. In the skin, psoriasis, burn, and so on. In the eye, in the joint, arthritis, and so on, kidney, and also all the multi-organ diseases like cancer, aging, diabetes, inflammation, infection. These all are the result of oxidative stress. Of course, oxidative stress is a very, very big problem because it toxifies the body. So where am I? Okay, just to move it and to go there. Okay. Usually in natural biological system, there are also many ways to combat the diseases, the oxidative oxygen damages. Every organism have many functional groups of compound with antioxidative activity. They are scavenging the radicals and preventing the diseases. For example, we have an enzyme compound like superoxide dismutase or catalase. We have small molecules like ascorbic acid or tocopherol, polyphenols like anthocyanin, polymers like melanin, and other lipophilic compounds. All these may reduce the problem of oxidative stress. For that, of course, it is very recommended to eat a fruit with a lot of antioxidant. This is a rule. People are eating a lot of antioxidant. From nutritional point of view, it is advised all the time to eat a lot of fruit. They have a lot, of course, the diet with fruit and vegetable are rich in antioxidant and can reduce different diseases. The thing is that it was found by many research that dark fruit and vegetable have higher antioxidant activity like fruit like blueberries, strawberries, grapes, and other fruit which are darker. The darker, the better. Okay, now I'm going to talk about medicinal mushroom. Medicinal mushroom, of course, have also antioxidant, and they are now considered as very good antioxidant source, similar to other colorful vegetables. We know I'm sure you know the Ganoderma, the Pleurotus, the Trametes, Cordyceps, and the Morel, of course. They are all, of course, as medicinal mushroom, they are used for centuries in the traditional medicine to prevent diseases, and they seem to be a very good source of antioxidant. If we see what they are capable to do, as we see in this summarizing slide, they have activity of many. They can solve or reduce the problem of many problems in the human body. They have a biological activity from antibacterial, antifungal, anti-inflammatory. As we see it, it's similar to the effect of oxidative stress. Here, the mushroom can prevent the effect of the oxidative stress, and they have a lot of antioxidant as well. Here we have a summarizing slide of the compound. Many secondary and primary compounds which have a biological activity and they can help to prevent diseases. If we are going to the antioxidant, mushroom are considered to be in between the very high, like the blackberries. Blackberries are very high with the antioxidant. Grapes, red grapes are also very high, but mushroom also higher than many other fruit and vegetable. For example, tomato is only half or less than half than the mushrooms. So if we eat the mushroom, we can get a lot of antioxidant activity. As we said, it can help in the health problems. The thing is, okay. The thing is about morel. I'm talking about morel. Morel is an edible mushroom, but it is also medicinal mushroom. As we see it here, morel have a very high nutritional value, especially in regard to the protein and also the fibers, of course, and the vitamins. Vitamin D, for example, is very high, iron, calcium, and so on. So it is a nutritional, also from the nutritional point of view, it's very important, but it's more known as very tasty, and its aroma is very high. That's why people are looking for it in everywhere. It's go over the world in the northern hemisphere, also in the Himalaya, in the Mediterranean, in Europe, and so on. And people are looking for it all the time because of its taste and its aroma, but it has a lot of nutritional value as well as medicinal value. Okay. So what kind of medicinal value morel have? Again, antioxidative, anti-inflammatory, anti-tumor, anti-parasitic, antibacterial, and so on. And we summarize it in one of our papers regarding the true morel with the nutritional and phytochemical composition, health benefit, and we see that morel have a lot of benefit. The thing is that there are so many, very high number of species of morel. Morel are divided to 3 groups. There are the black morel, the yellow morel, and there is a group of white morel. And the question is, are they the same? Do they have the same quality as in regard to the antioxidant activity and the beneficial qualities. If we look here, we see the one example of morel, Morchella esculenta. We have many compounds, of course, and their biological activity. For example, phenolic compound may have antioxidative activity, antibacterial, anti-allergenic, anti-inflammatory, and anti-tumor. Polysaccharide of mushroom have a lot of antioxidant activity as well, and so on. Tocopherol, also antioxidant, organic acid, also antioxidant. So it seems, again, that the mushroom can be a source of antioxidant, very good source of antioxidant. As said, there are many species, and they grow in different habitats. The question is, are there differences between mushrooms that grow in different environmental places? For example, in a paper that was published at 2013, comparing the same species that grew in different places, some in Portugal and some in Serbia, we see that there is difference in the quality of the mushroom and also in the scavenging activity, where in Portugal it was higher than in Serbia. That means that the local environmental condition may affect the quality of the mushroom and also their beneficial effect on the health, of course. In our study, we looked at two species. One is yellow morel and one is black morel. And we found that within the population, we have also differences in the color of the mushroom. Even if it's known as black morel, in some cases we have other colors, like here we have the brown, we have the white also sometimes in the same population. And here this was the Morchella conica, it's black morel, and here we have the yellow morel, but the yellow morel is not all the time yellow. Sometimes it's also brown. And the question is there also differences between these two species and within the species itself, within the different colors of the mushrooms? So what we have done, that we took different mushrooms with different colors from the same population. It was grown in the same place. And we have done the powder, which was then examined for the antioxidative activity. As you see it here, all the extracts differ. They are the same mushrooms, the same population, the same species, the same site also. So what we have found that the two species that we were looking at differ in the color nuances, but as you see it here, this is Morchella esculenta, the yellow morel, and this is the conica, the black morel. Here, this is the color nuances that was measured by Minolta chromometer, which can analyze the color in different nuances. For example, the L parameter is the darkness. The A parameter is the green and red. The B parameter is the blue and yellow. C is intensity. And H is predicting the total color of in this case, of the powder. So it was very surprising that although there is, for example, from the point of view of the color, there was difference between the yellow and the black morel, it was not significant. Also, we are talking about black and yellow morel. Although there is difference, it was not significant. Similar in all the other parameters of the color. That means that the population itself have range of colors. And the question again, as we said, is it matter? Is the color matter? will darker or lighter, mushroom will be better as in the case of antioxidant. The antioxidative activity was measured by DPPH. It's measuring the radical scavenging activity, where the DPPH can interact with the antioxidant and change the color from purple to yellow. So this is the measurement. So we could add this compound into with the powder of the different mushroom, and to see if there is any differences in the antioxidant activity. We checked also the phenolic compound and the antioxidative activity, as well as the colors of the two population, of the two species actually. And what we have seen again, that the black mushroom had higher color, had a darker color, of course, but not significant because of the diversity of the colors within the population. And also the phenolic compound and the antioxidative activity differ, but again, not significantly. So that means that although there are two different species, there are not, there is no difference, significant difference between the species, but within the species. Here we see the correlation between the radical scavenging activity and the phenolic content. Of course, in this case, there is a correlation between the two, and it's significant correlation between the antioxidant activity and the phenolic compound of both species, the yellow and the black morel. So phenol play an important role in the antioxidant activity of the mushrooms. And here we see the correlation between the antioxidant and the color. What we see here that while the black triangle are the esculenta, which is the yellow morel, the yellow morel has higher correlation between the color and the antioxidant activity. While the black morel, it's much lower correlation, which means there was almost no correlation between the antioxidant and the color of the mushroom. So it seems that in the case of the black morel, darker morel were not so much higher in antioxidant activity. The population was similar, while in the yellow morel, the color make the difference, and the darker color got higher antioxidative activity. Okay, this was the summary of the talk, and the point is that if we are looking of a better source for the antioxidative activity, we need to know better what kind of mushroom to take. Of course, when we are taking a lot of mushroom with the whole population, maybe there is no differences, but within a population, there might be differences in what kind of mushroom we are taking. The darker was better in the yellow morel, but it was not better in the black morel. Other factors may affect, of course, the antioxidant beside the color. The certain site, the chemical in the soil, the temperature, many other factors, environmental factors that can affect the mushroom. But if we are looking about the color, as I said, in some species it was correlated, and in some species it was not correlated. And of course, we are continuing now to look for other correlation effect of an environmental factor on the antioxidant and on the other beneficial activities of this mushroom. And I will finish with the acknowledgement of Limor and Gorinaz, which helped me with this work. And that's it. If you have any question, I will be happy to answer.